This video will focus on gene interaction, and some of the topics that we'll focus on are shown up on the screen. Let's begin with a few examples of variation in which alleles can be expressed. So first of all, let's begin with simple dominance. This is one that you're most familiar with. This is simply we have one allele that tends to be expressed over another allele. So you can think of all sorts of examples of simple dominance. In this particular case, here's a situation if we're looking at an allele that codes for the enzyme phenylalanine hydroxylase, or PAH. And it turns out that some individuals who are homozygous recessive, as we see on the far right, are unable to produce any functional enzyme, and therefore they're considered phenylketonurics because they have a difficult time processing phenylalanine, which is a simple amino acid. However, other individuals who have at least one dominant allele are able to produce enough functional enzyme. Another example of variation in alleles is incomplete dominance. So here's a situation where if you cross two individuals, looking at the lower left panel here, homozygous dominant times a homozygous recessive, notice that the heterozygotes tend to have a blending of the two original phenotypes. So we might say in this case the phenotype is dose dependent. And again, this is an example of incomplete dominance because if you look at the phenotypes, it's not really clear which is dominant, the red color flowers or the white. On the other hand, we can have a situation referred to as codominance. So the ABO blood types are classic examples. So here I've listed genotype possibilities for the 4 different possible blood types down here on the lower part of the screen. And notice there are 2 versions of this allele that codes for the proteins on the outer surface of red blood cells, the recessive version indicated by small i's, and 2 dominant versions, the A version and the B version. And so again, this is considered codominant because if a person has both the A and the B alleles, they produce both the A and the B proteins. And again, that's a little different than incomplete dominance because in this situation, we have both the A and the B being equally dominant. There's no real blending of the two. They're both equally expressed. On the other hand, there are situations where certain alleles are lethal when found in homozygous recessive conditions. Just as one example, there is an allele that codes for fur color in mice, for example, and it turns out that that allele has pleiotropic effects. So not only does it code for coat color, but it also it turns out is recessive lethal. So those mice that unfortunately end up being homozygous recessive for this particular allele, they end up dying. And so they just don't show up in the population. So if you do a simple cross between 2 heterozygotes, the heterozygous condition codes for yellow fur. The homozygous dominant condition codes for black fur. If you do a simple cross between 2 heterozygotes, so you'll see sort of four possible offspring. It turns out one of those offspring, again, the homozygous recessives, tends to die off in the population. So you end up with a 2-to-1 ratio of live offspring instead of a 3-to-1 ratio, as you would normally expect from a simple cross between 2 heterozygotes. Let's briefly review the ways in which genes can sort of interact with each other and the effects that they can have on phenotypes. We have seen this before. First of all, this concept of pleiotropy. We're talking about situations where a single gene can have more than one trait, and there are all sorts of examples. For example, the gene that codes for a hormone receptor, such as a progesterone receptor. If you were to knock out that one gene, in mice, for example, you can see multiple different effects, like we see toward the lower right-hand portion here. So that could affect the uterus, the ovaries, mammary glands, and so forth. So the point is that's a situation where one gene affects more than one trait. We can find similar situations even in plants. And so it turns out in plants, one gene controls both the length of the corolla tube, that's just kind of the base of the petals here, as well as the filament. That's the structure that produces the anthers at the top. And of course, anthers produce the pollen. So the point is, these 2 structures are under the control of one gene. On the other hand, again, we've seen this before as well, polygenic effects, traits that are affected by more than one gene. So height is a classic example. We see sort of essentially a normal distribution if we look at height of individuals. And again, this is due to a variety of factors, but one of which is the fact that height is controlled by more than one gene. Likewise, if you do studies of stress in animals, mice for example, if you put mice in an unfamiliar environment, they get a bit stressed out, and it turns out there are at least 3 genes that are expressed that play a role in the stress response in mice. So again, another example of a polygenic effect. So this slide just sort of summarizes pleiotropy and polygenic effects. So pleiotropy, again, one gene affecting multiple traits. Polygenic, multiple genes affecting a single trait. Let's now look at the ways in which genes can interact with each other. Let's begin with corn snakes as an example. So in this example, we're looking at genes that can act totally independently of each other to influence a trait. So here we're looking at genes that have an impact on the coloration of corn snakes. So if you've ever seen corn snakes, they can come in a variety of colors. Top left, this is camouflage. These are snakes that produce both the orange and black pigments. So some snakes are camouflage, some are orange, some are black, and some are albino. And so there are 2 genes controlling the coloration of skin in corn snakes. And so if you do a dihybrid cross of 2 individuals that are heterozygous for both of these genes, you get a typical 9:3:1 ratio. And so let me just quick preview. So you can use whatever letters you want to keep track of these things, but let's say the O gene codes for orange pigment. So if you have at least one dominant O, you're going to produce orange pigment. Let's say the B gene codes for black pigment. So again, at least one dominant B gene, you'll produce black pigment. If you produce both the O and the B dominant alleles, you'll have a camouflage snake. But if you're homozygous recessive for both of those genes, then you'll have an albino. So again, if you do a typical dihybrid cross of 2 heterozygous individuals, so we're talking about this line right here, the F1, where we see camouflage, again, heterozygous at both the O and the B alleles, you get your typical 9:3:1 ratio, 9 camouflage, 3 orange, 3 black to 1 albino. And again, what does this tell us? Well, this 9:3:1 ratio tells us that these 2 genes, in this case the O and the B, they act independently of each other. They really don't interact much at all. I mean, they both play a role in skin coloration, but again, they both act independently of each other. Let's look at other examples. In this case, complementation. So we're going to look at harebell flowers as an example. And here we have a situation where you have 2 genes that impact the coloration of these flowers, and it turns out to produce blue flowers, you need a dominant allele at 2 different genes. So a dominant allele at both loci. The other option is if you don't have that situation, then the flowers are white in color. So here's the situation in these harebells. Again, to produce blue flowers, like we see toward the right here, there are 2 colorless precursors that are produced, so just 2 molecules that are colorless. So colorless 1 is converted to colorless precursor 2 as long as enzyme 1 is present. Colorless precursor 2 is converted to the blue pigment as long as enzyme 2 is present. So each enzyme coded by a different gene. So again, you need at least 1 dominant allele for both the enzyme 1 gene and the enzyme 2 gene in order to pass all the way through this pathway to produce the blue pigment. If you're lacking either enzyme 1 or enzyme 2, as we see toward the left here, you can't produce the blue pigment and the flowers will be white. So if you do a dihybrid cross of 2 individuals that are heterozygous for both of the alleles for those 2 different enzymes, and again, you can use whatever letters make sense to you. Your book uses W1 for the enzyme 1 allele, W2 for the enzyme 2 allele. So again, we're going to jump right at this line right here. We're crossing 2 individuals. Again, heterozygous for both of those 2 different genes. So the plus indicates the dominant, with the lack of plus indicates the recessive version. We get our 9:3:1 genotypic ratio. as we typically would expect for a dihybrid cross between two heterozygotes. But if you look at the phenotypes, we come up with a 9-to-7 ratio, so 9 blue to 7 white. And again, if you do the cross yourself, recall that you need at least one wild type, in this case ended by the plus, for each of the two different genes, the W1 and the W2, to produce the blue pigment. If you're lacking either of those, then you end up producing white flowers. So again, this 9-to-7 ratio, very characteristic of this phenomenon referred to as complementation. Another example, there are situations very common in plants where genes are duplicated, and sometimes they do the exact same thing as the gene from which they were copied from, and sometimes they can be modified to take on different roles. But let's look at an example of duplicate genes here. So in this situation, you might have two genes, but you only need one dominant or one normal allele to produce your normal phenotype. So as an example, there's a situation called narrow sheath in maize, illustrated down here in panel B. And so these individuals, if they're homozygous recessive at these two different genes, again, you can use any letters you want, but by convention for this gene, NS1 and NS2, the two narrow sheath 1, narrow sheath 2, if you're homozygous recessive at both of those genes, then they'll produce the narrow phenotype. So, if you have at least one allele, then they produce the normal phenotype. So if you cross two individuals that are heterozygous for these two genes, what type of phenotypic ratio do you end up with? Well, again, let's cross two individuals, heterozygous, as we see in this line right here, for both of the genes. Typical dihybrid cross, again, your typical 9:3:3:1 genotypic ratio. But then if you look at the phenotypes, again, all it takes is one dominant allele to produce normal leaves. So you end up with a situation where you have a 15-to-1 ratio. So 15 of your offspring will produce normal leaves, and only one, that one which is homozygous recessive for both of those genes, will give you the mutant or the narrow sheath mutation. Let's look at another example, this idea of epistasis. One gene has an impact on the expression of another gene. That's epistasis in general. So you might say one gene suppresses the effect of another gene. In this case, we're going to look at recessive epistasis because we're looking at a situation where a recessive genotype controls or sort of dictates the overall phenotype in question. So the recessive genotype or phenotype overrides the expression of a totally different gene. So in this case, we're going to look at Labrador retrievers as an example of recessive epistasis. So you think about Labrador retrievers, they typically come in three colors. You can have chocolate or brown labs, black labs, or yellow labs. And so again, in this case, we're looking at fur color. In this case, it's controlled by two separate genes. Again, you can use whatever letters you want, but typically we use the E and the B genes. And so let's first of all look at the E situation. If a dog is homozygous recessive for the E gene, they'll produce yellow fur regardless of the situation at the B gene. If the individual is homozygous dominant at E or heterozygous at E, that'll indicate dark fur. It says nothing about whether it's black or brown, it just says it can produce dark fur. Now, again, on a totally separate gene, namely the B gene, if the individual is homozygous dominant or heterozygous, that dog will be black. Again, as long as there's at least one dominant E. On the other hand, if the dog is homozygous recessive for the Bs, it will be brown, again, as long as there's one dominant allele for the E gene. Okay, so that's the basic setup for recessive epistasis. So again, if you do a cross, typical dihybrid cross two individuals that are heterozygous for both the B and the E genes, what you end up getting is something like this. So here we see a Punnett square of the cross I just mentioned. Again, cross two individuals heterozygous for both of those genes. You end up with a typical 9:3:4 ratio. So in this case, you would have 9 black labs, 3 brown labs, and 4 yellow labs. So that ratio, 9:3:4, again, very characteristic of recessive epistasis. On the other hand, we could talk about dominant epistasis. So where a dominant genotype or phenotype overrides or controls the expression of a totally separate gene. So in this example, we'll look at foxglove flowers. So they can come in various colors, even more than what's illustrated here, but common colors for foxglove can be white, light red, or dark red. And also notice these spots in the throat or in the middle portion of these flowers. Keep that in mind for a few seconds here. So let's set up a situation where we're looking at the control of the color of the petals of these flowers. So again, under the control of two different genes, W and D. And first of all, if you have a dominant W, that's going to produce flowers that have coloration only in the throat spots. It won't produce any coloration in the petals. You end up with a white flower. On the other hand, if you have a small or recessive W, then that will allow coloration to be produced throughout the petal. Okay. What controls whether it's light red or dark red? Well, a totally separate gene, namely the D gene. So dominant D gives you dark red, small d gives you light red. Okay. Again, as long as you have a small w in that other gene. So again, if you do a cross between two heterozygotes for both W and D, you end up with again, your typical 9:3:3:1 genotypic ratio, but if you look at the phenotypes, you end up with a 12:3:1. So 12 of your offspring are white with spots in the throat, 3 are dark red, and 1 would be light red. Again, that would be the one that is homozygous recessive for both the W and the D genes. So again, this 12:3:1 ratio very characteristic of dominant epistasis. So this slide just summarizes the situations we've discussed so far. So no interaction between two genes, your typical 9:3:3:1 ratio. That was our corn snake example. Complementation, this was our harebell example, 9:7 ratio. Gene duplication or duplicate genes, this was our narrow sheath in maize, 15:1 ratio. Recessive epistasis, this was our Labrador Retriever, 9:3:4 ratio. Dominant epistasis, 12:3:1 ratio. Few other concepts to round out this chapter. Variation in which phenotypes can be expressed. So first of all, the idea of penetrance. If you have some individuals and you know their genotype, let's say, for example, you're looking at a certain tree, and if you have a gene that codes for leaf color, and if you know that homozygous dominant, in this case G, or heterozygotes should produce, let's say, green leaves, whereas homozygous recessive produce, let's say, yellow leaves, if you look at those individuals that are homozygous dominant or recessive, what you might find is that not all of those individuals produce the green leaves as expected. So in this case, there's not always a clear linkage between genotype and phenotype that we see. So if in this situation, if you see, well, only about 70% of the individuals express a phenotype that should express a phenotype, you might say, well, that allele, that gene is about 70% penetrant. Again, because it doesn't always produce the phenotype that you would predict in the offspring. And there are various things that can contribute to that, such as there could be other genes involved, there could be environmental factors involved as well. Okay. Let's just jump on to the next concept, namely expressivity. And so let's say, for example, you have an individual, let's say you have a plant, again, let's say it's homozygous dominant for some gene that codes for green leaves, and it turns out if you look at different individuals, yes, maybe they're all producing green, but in different levels, right? Different variations of the extent to which green is produced. Likewise, if you look at piebald spotting in dogs, common in many dogs. So if you look at the spots produced on the fur, under the control of one gene, and so you might see a bunch of dogs that have the same genotype, and yet they have radically different extents to which they have this piebald spotting in their fur. So again, you can say the expressivity of that particular gene or allele is highly variable. That concludes our brief overview of gene interactions.